So yeah, hey folks, welcome to the last special topics lecture. It's my excellent pleasure to introduce Professor Yatish Turakhia. He's a professor in the Electrical and Computer Engineering Department at UC San Diego. Yatish and I have done a lot of work in the past with kind of large-scale evolutionary trees. So all of you did a little bit of work inferring evolutionary trees of SARS-CoV-2 and HIV. And Yatish's lab does a lot of work kind of building those methods and making sure that they're fast enough to be able to run on these massive data sets that we're generating. So yeah, let's welcome our guest speaker. Thanks for being with us, Yatish.
Thanks, Nima, and look forward to the interaction as well. So today I'll be presenting some of the work on that we've been doing on phylogenetics for genomic surveillance. So I'm not sure if you guys are familiar with all these terms, so I will introduce it. Probably Nima has already introduced some of these terms, but I will introduce it anyway. But before I do that, maybe I'll give a quick introduction about myself. So I did my education mostly in electrical engineering, starting with my undergrad in India at IIT Bombay, where I did both my bachelor's and master's in a sort of a co-term program with specialization in microelectronics and a minor in computer science, during which I had the good fortune of also interning twice in the summer at Carnegie Mellon, where I worked with Professor Diana Marculescu and Siddharth Garg. And then I went for my PhD at Stanford, where I was again very fortunate to be advised by two great people, Professor Bill Daly, who's also a chief scientist at NVIDIA, and Professor Gil Bajarano, who's a professor in pediatrics and developmental biology. And during my time there, I got to work at a number of firms, Apple, Qualcomm Research, Microsoft Research, and had a great time doing research at the intersection of computer architecture and bioinformatics. Um, and then I went for a short postdoc at Genomics Institute at UC Santa Cruz, where I was joined between David Hausler, Benedict Payton, and also Russ Corbett-Datek. Russ is the one who I worked closest with during the time of the pandemic and since then on various tools that I'll be presenting today. And then since 2021, I've been at UC San Diego as an assistant professor in electrical and computer engineering. Um, and at UCSD, I sort of lead a interdisciplinary lab where we work at the intersection of algorithms, biology and medicine, and computer architecture with this broad mission to develop parallel algorithms, software, and domain-specific hardware accelerators to enable faster and cheaper progress in biology and medicine. So with that said, maybe now I should start introducing like the basic terms that I'll be repeating over and over again throughout this talk. And the most important of that is the term of the genome. So very broadly, this is basically just a sequence of letters that are contained in your DNA, which make up your genome. But that's basically what forms a program or the blueprint of life for all life forms, right? Um, and even viruses have a genome, even though it could be debated whether they are a life form or not. But even they carry a genome, and that's what I'll be referring to later as well. But essentially, you can think of it like a program which is encoding for a certain output. In our case, it's encoding for various physical characteristics that we might have, like two eyes or hands, legs, all of that. There's some sort of a recipe in this particular program, which is the sequence of the DNA, which then make up the genome. And genomics is basically a field that relates to the study of that genome, right? So it's basically a lot about studying these genome sequences, right? Um, and what's important is that in with modern day technologies, it's actually possible to read out the sequence that a sequence of letters that's contained in the genome. It's typically consisted of an alphabet of just four characters, A, C, G, T, in case of the DNA. And we have technologies called sequencing technologies that allow you to, take a sample from any living organism, could be like a saliva or blood sample, and these machines can actually read out what are the DNA sequences contained in them, right? And this is a very powerful tool, especially in the context of molecular epidemiology, which is sort of the theme of this presentation. And so to introduce that, I think maybe I should highlight this picture first, which is about the evolution of the COVID-19 virus. We call it SARS-CoV-2 in the scientific naming convention. But as well, all of you would know that this is a virus that actually mutates. And what we mean by mutation is that its genome sequences is constantly changing due to various factors, right? So as it's spreading from one individual to the other, or as it's replicating within an individual itself, it can acquire some mutations, right? So this is what this figure is showing, that initially, let's say you had like this virus with a blue genotype, and then it replicates. It may or may not mutate, but during some replications, it may acquire some mutation that changes its form, right? So it's these mutations that make the virus continuously change its form. And because of that, it may acquire certain new physical characteristics which might help it better replicate or infect new individuals, transmit better or faster, survive under certain conditions, but also be more lethal in some cases, right? So that's what this is saying. Certain mutations may render the virus more contagious, virulent, or capable of even evading vaccines and certain antibody-based therapies that we have developed. So basically, the technology that I just mentioned, genome sequencing, that's the technology that would help us to monitor how this virus is changing and its different evolutionary dynamics as well. Um, so for that, let me now introduce the other term that I used in the title of my in my title slide. That's that of phylogenetics. So to that, let's just look at this picture once again, which is that of the virus replicating, and while replicating also mutating and changing its form, right? So we don't really get to see this picture, right? Because we are not in real time seeing how this virus is developing and its many copies and what sort of sequences uh, it contains. Rather, what we are able to do is that when a person gets infected, that person can go to like at the clinic, that person can get sequenced, which basically means that we collect or we read out the genome sequence uh, that makes up the virus that infects that individual. So that's what we could do, and we could do this for many individuals. In this case, I've shown an example of 6 individuals labeled A to F. And from there, we might be able to infer like how these different sequences differ from each other, right? So let's say I call A as my reference, then B has like one mutation compared to that reference, right? C has no such mutations. D again has one mutation. E has two, and F has one. But some of these mutations could be very different, right? Even if they have same number of mutations, they could be actually different mutations altogether. So what we mean by phylogenetics is basically taking these genome sequences that we might have collected from, let's say, clinical sequencing data, and then trying to infer like a family tree out of it. Basically, what that means is that the sequences that you've collected appear at the tips of this tree. And on the branches of the tree, you might infer the mutations that might have occurred. So, for example, the starting sequence might be similar to that of A. And as it replicates, it acquires this yellow mutation and green mutation. So E has both yellow and green, D has only yellow, B has yellow, and so on. But effectively, what this phylogenetic tree is doing is it's grouping similar sequences together, right, in the tree. And the more dissimilar sequences are more far off in the tree. So it's sort of like constructing a family tree, which sort of represents the transmission dynamic pattern that you see on the left. Not exactly, but sort of from this particular tree, you can actually start making inferences about the transmission patterns pretty accurately as well. So that's basically what I mean by phylogenetic inference from genomic sequences. And these trees effectively form the basis of various evolutionary and transmission dynamic studies of this particular pathogen, right? So this was very widely used, for example, in the pandemic, and even now it's being quite uh, prevalent, although the sequencing rates have decreased, but we still try to keep monitoring how the virus is evolving so that we can respond to it effectively. So with that said, maybe I'll quickly provide an outline of this talk. I'll first introduce like how during the pandemic we saw a flood of genomes, and then the tools that we developed, primarily Usher and the tree that we constructed using this particular software for SARS-CoV-2, and then the applications of this tree, and then what else are we working on. I should also credit Angie Heinrichs at UCSC, who actually developed most of these slides. I decided to use some of her slides mostly because of like how well designed they were for a very broad audience, right? Um, so just to give you an idea of like how much data was collected during the pandemic for SARS-CoV-2, maybe I'll take an example first from another viral sequence called some viral species called RSV, which also infects lots of individuals in the US and is responsible for many flu-like symptoms in individuals. And if you look at how many sequences we've been collecting over the years for this pathogen, that's been shown on this particular plot. And as you can see that it's risen quite a lot over the years, especially in the last decade or so, as the sequencing technologies have advanced, both in terms of cost and throughput. So we have thousands of sequences these days on like public databases like INSDC. But just compare that to what we saw for SARS-CoV-2, the COVID-19 pathogen, and you will see that the scales are totally different. In fact, RSV gets completely flattened if you try to visualize this on a linear scale. And as you can see, we had seen like a complete exponential rise of SARS-CoV-2. It in fact is the most sequenced species on Earth today, with about 16 million sequences already sequenced and deposited on public databases. Uh, so the point I'm trying to make here is that while we did have a lot of tools to analyze genome sequences in the past and estimate phylogenetic trees, they were just not designed to handle the scale that we witnessed because it was quite unprecedented during the time of the pandemic. Um, and just to give you an idea about that, there were several days during the pandemic where we saw tens of thousands or even hundreds of thousands of sequences being deposited uh, onto these databases. But if you look at what the phylogenetic inference tools were capable of, for example, how many sequences could they handle in a single day? It would be around a few thousands at most, right? Um, so they were just not capable of building trees which would comprehensively handle all the sequences that existed on public databases. And that's the sort of problem that we wanted to tackle with Usher. And later I'll tell you why we wanted to build comprehensive trees rather than just subsample and build trees for a smaller number of sequences. Those kind of aspects I'll cover later. But for now, I'll just say that that problem is important to build trees which are comprised of all sequences that are available, right? So for that, we build our tools called Usher. And I'll now tell you what Usher essentially does. So um, the key idea here is that we want to build, like I said, a phylogenetic tree of different SARS-CoV-2 sequences, where the sequences that you've collected clinically are actually represented at the tips of this phylogeny. And so this is an example of how that would look like. And I'm talking about the early days of the pandemic, March 2020, where we already had thousands of sequences onto the trees that people had constructed back then. But now when you have a new sequence coming in, let's say a person got infected and was newly sequenced in a particular clinic, you want to try and accommodate that sequence onto the tree just to see what its context is, right? Is it similar to the sequences shown here, which are shown in yellow and green, or something similar to the ones at the bottom shown in blue, right? And that sort of helps us infer like which parts of the tree are evolving faster, which strains are evolving, many of those applications. But you sort of want to contextualize it. What is this particular sequence most closest to? Where would it fall on the tree if I accommodate it, right? So that's basically what Usher is solving. And the idea of the software was that it's supposed to be ultra fast in the sense that it's supposed to accommodate new sequences that emerge in real time, which means that you collected the sequence and within seconds to minutes, I actually accommodate it on the new tree and can do various sorts of analysis on that, right? Uh, so in order to make this so fast, we obviously had to change the paradigm of how we build these phylogenetic trees, right? So one of the key tricks that we've used is that rather than uh, building an alignment, basically sort of like identifying correspondence of characters between all sequences that are collected, including this new sequence that existed, which is what people would do for estimating these trees. Usher would actually just use the sequence itself and its comparison to a reference to accommodate that new sequence onto the tree. And essentially what it did very broadly is that it introduced a new data structure called mutation annotated tree, which had all of this information already contained in it, in the sense that it had one sequence at the root of the tree, and then on the branches of the tree, it basically annotated mutations that have been inferred to have occurred on those branches, right? And secondly, what it did was it actually used an approximate technique called parsimony, rather than using more like elaborate techniques or compute intensive techniques like maximum likelihood for finding where should that sequence be accommodated, right? Now, I should say, even though this technique is approximate, it actually works pretty well for pathogens like SARS-CoV-2. In fact, it's nearly as good in accuracy as some other techniques. We've shown that, mostly because the rate at which we were sampling the pathogen, we would on an average see that the newly sequenced sample that you've collected often differed by one or fewer mutations on average compared to the closest neighbor in the tree, which basically means that you can pretty confidently place it using approximate techniques as well, like maximum parsimony. And finally, another reason that Usher was so fast is because we actually parallelized it. So we used our skills in parallel computing techniques in the lab to make sure that, for example, utilize many CPU cores that would be available on your computer, and make sure that the processing gets done as quickly as possible. And then, like I said, that the paradigm shift that we did was that we don't rely on this whole MSA of all sequences. Rather, what we did was we compared it to a single reference and then tried to accommodate that new sequence onto the tree itself. But more importantly, we also made sure that we didn't infer the tree de novo, which basically means that we don't collect all sequences and then estimate the whole tree from complete scratch. Rather, what we do is that we take the tree that we inferred for the previous day, and then just try to accommodate the new sequences onto the tree without disrupting the relationships that you already inferred in the previous tree. So that was the main idea. Anyway, those were just some technical details, but now I'll tell you more about like how people used Usher in practice. Now that we've enabled the speed that we did, where you can actually literally place it in real time. So the one thing that we did was, and this is again credit to Angie Heinrichs at UCSC, she actually developed a web interface for Usher, as I've shown here, where people could literally upload their sequences onto this interface and get the new sequences immediately accommodated on the tree. And here I'm showing you an example of how that would look like. Obviously, this is zoomed into a very, very small portion of the tree. Remember, the tree actually consists of thousands of sequences, actually millions of sequences. And we've actually zoomed into a region where this new sequence got placed. And I'm showing you like 6 or so neighboring sequences around it. But this is what people would be most interested in. So this is, for example, the new sequence that just got placed. As you can see, it's from California over here from this identifier. And as you can see, it's actually getting placed next to some of the sequences which were collected in New York, Utah, and so on, right? But this is the main idea, that like how people would use it, and that would typically that the most common use case of this was to be able to name new variants of or identify and name new variants of this pathogen, right? So here's an example of how that is done. So what people would do is they would, whenever they actually find new sequences, they would place it using Usher's web interface onto the Usher tree, and then contextualize those new sequences in uh, 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 in the presence of all previously sequenced samples. And based on that, they would actually name the lineage of the new sequences, right? So in this case, for example, what people have done is, it's actually the way the new variants get named is using GitHub issues. So you can actually visit this particular GitHub repository and see how people are actually naming lineages. All of this is happening in full public view. But the idea is that this person has actually discovered potentially a new lineage using 52 sequences, as they have mentioned, mostly collected from Singapore. And those sequences have been placed with the help of Usher, and this is that zoomed view that I told you about. Um, and they report that this is a sublineage of XBB.1.5. Essentially, you can think of this lineage name as being an address on the phylogeny, right? So what this means is that it's there's a lineage called XBB whose descendant lineage is XBB.1, and within that, there could be a sublineage 0.5. And now these 52 sequences fall as a sublineage to this particular lineage, right? So it's sort of like an address onto that tree. And these are the new mutations that could have been acquired by the sequences on top of whatever XBB.1.5 contained, right? So that's the idea. And then there would be reviewers uh, 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 who would do some quality controls and checks on whether this particular subtree looks like a true new variant. And if that is the case, then they would actually designate it officially, right? And that's how new lineages would get named. And in fact, I should share one story which I found very inspiring. Actually, even the Omicron variant was discovered in this way. So Omicron's scientific name is lineage name is B11529. That's the first, like the root lineage that formed Omicron variant. And essentially, the way this was discovered was by the scientist called Tom Peacock from Imperial College in the UK. Essentially, what he did was he noticed that there were four sequences that were collected and newly deposited in public databases back then. Three of them belong to Botswana, and one of those sequences came from Hong Kong, who was a who was from a from a sample who was a traveler from South Africa. And this particular sequence was partial, so that's why it was actually not placed. But the three complete sequences were placed on this tree, and they are shown as these three tips highlighted in red. And what you would find interesting is that the context of these three sequences is that it's forming a really long branch, as you can see over here, onto this tree, right? This is a new branch that these three sequences form. So they are all related to each other, very closely related to each other, share many common mutations, and they fall on this branch of that tree, right? That's kind of the idea. And if you look at where this particular subtree falls, it's actually falling within this lineage, previously existing lineage called B11. And the reason why this branch is so long is because there were many mutations that were acquired by these sequences on top of this lineage B11, right? So what the scientist is saying on this public post, the GitHub issue that I mentioned earlier on the repo that I showed, that this seems to be like a complete new lineage with many mutations that have been listed over here. And several of these mutations we knew already were problematic in the sense that they would confer virus with new abilities to transmit and infect individuals. And so they have proposed that this be named as a new lineage. And in fact, people reviewed this very quickly. It was urgently reviewed because of the mutations that this particular strain had acquired. And within a day, it got named this sublineage called B11529. And within 3 days after that, WHO actually called it Omicron, the variant of concern, because I think scientists all over had noticed that some important new mutations had been acquired and that needed to be tracked quickly. So that's how Omicron got named. Another story which I find quite interesting is how people used Usher to actually analyze outbreaks in local areas. And this particular example comes from like an elementary school in California, where students and many parents of those students were infected with basically the Delta variant, which was prevalent at that time. And in fact, till that point, it wasn't clear whether little children could, infect or actually do onward transmission of this particular pathogens and act as transmission agents to adults. But this was the first such recorded among the first such recorded cases, where it became very clear that the transmission pattern sort of looked like the potentially the teacher infecting the student and then the students acting as agents and then infecting the parents. And for this, using those trees was an important criteria where people could actually trace when new mutations got acquired and which were the sequences that might have arisen earlier than the others, and so on. And so this particular article was in fact it in fact appeared in the New York Times, where uh, uh, because of its significance, where we first found that an adult could infect children, and in some cases the children could then go on and infect the adults again. So that's about some of the applications that I mentioned related to Usher. Uh, One particular problem that we've obviously encountered with the sheer volume of the data that was produced in the pandemic is how do you keep growing the tree, right? Because you have so many new sequences that are continuously being acquired. What do you do to make sure that you can uh, catch up to the volume of the data that's being generated every single day? So here's what I'm showing an example of, like how quickly the data was growing. So this is like a y-axis in millions, and as you can see that it's actually growing very, very fast, especially 2021, 22 time frame. It grew pretty fast. Now it's actually reduced since the pandemic has been declared to be over. So it's kind of saturated at a little over 16 million for many months. Um, but this is how quickly the data was growing. And somehow we had to build trees which had roughly the same number of sequences as the database itself. Now you can see that there's a small gap that exists between the number of sequences in the Usher tree versus what we see on public databases. And that gap is mostly for quality control reasons. We had to start filtering out few sequences because they could not be confidently placed on the tree, or they had some quality issues, which is why we decided to exclude them intentionally. So that's why the small gap exists. But pretty much Usher was able to catch up to the amount of sequences that existed publicly, right? So how did we do that? There were two aspects to it. One is that we used Usher to place those sequences. But because Usher used a greedy strategy, which means that it didn't allow relationships of existing sequences to be disrupted when placing new sequences. But that's not a fully optimal strategy. So you have to somehow, like, sometimes refine the tree as well, because you might have made mistakes in placing new sequences. So there's also software we developed called Mat Optimize, and that actually was led by an undergrad student in my lab called Cheng Ye, was pretty brilliant in parallel programming skills and also algorithmic skills. But he developed this software which was several orders of magnitude faster than what previously existed, which could rapidly optimize this tree and fix potential mistakes that we might have done through Usher inferences. So this also needed to be done alongside Usher. Um, and of course here also we gain efficiency mostly because of the parallel uh, 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 programming techniques that we used. In addition, we had to support many users who wanted to do downstream analysis once the trees were inferred, right? So Jacob McRoom, who was a PhD student at UC Santa Cruz with us, he actually developed this utility called MatUtils, which is basically the mutation annotated trees toolkit where people could do various important functions like extract certain information that they would care about, like the variations or the tree subtree structure, or annotation of lineages within the tree and so on. So many important structures. But of course, at some point, as you would have guessed, with the sheer volume of data, we actually started encountering challenges. So what I mean is uh, that the number of new sequences that were being deposited, especially in from mid 2021 to mid 2022, we found that on several days it took us longer than a day to accommodate those sequences onto the tree. As you can see, the spikes are pretty high. Uh, a single day sequence sometimes would take 50 plus hours, which basically means more than 2 days to place it. What that meant was that we were at the risk of falling behind uh, the rate at which the data was growing, right? And that had to be fixed. So the first version of Usher, even though it was super efficient, was just not fast enough to be able to catch up to the scale. And again, Cheng, who developed Mat Optimize earlier, he helped, uh, further improve the efficiency of Usher, and we developed a new software called Usher Sample, which was even more efficient at placing it. And it was so efficient that since then till today, we've never encountered even a single day where that day sequence took us longer than 24 hours to place them. In fact, it's been substantially lower. In most cases, just less than 15 hours, or actually more like 5 to 6 hours on an average. Um, and the reason why this particular program is even more efficient is because, again, we use parallel computing techniques where we more efficiently parallelize how we placed multiple independent samples. We reduced many traversals in the tree, which were kind of redundant. We also used vector instructions, which is another form of parallelism that we exploited to implement this particular software. Um, so that's about Usher sample. Maybe I'll pause for a few seconds just to see if there's any questions, Yanima. Or
Yeah, any questions? Feel free to type in the chat, feel free to unmute. All good so far?
Yep. So maybe I'll move on.
Perfect. Yeah, go for it. Go for it.
Okay, sounds great. So again, just to go back to some of the applications, besides the few which I mentioned earlier, uh, so this Usher, because it was so efficient but also very accurate at incorporating new sequences onto the phylogenetic tree, we actually incorporated it into the official system for naming lineages. What that meant, or sorry, assigning lineages. This is a tool called Pangolin, which then integrated Usher in its default mode. So the idea here is that when a clinic, for example, collects new samples, they want to know whether the sample that they collected belongs to, let's say, Omicron versus Delta. Then what they would do is they would place it onto the tree, and then from the tree itself, you can actually tell whether it's a lineage called BA1 or BA2 or whatever, right? As a lineage of Omicron or not, for example. Um, and that became pretty efficient. And prior to this method of using tree placement to guide the assignments of lineages, people used to use machine learning techniques and other such approaches, and they tended to be more noisy than this phylogenetic placement approach. So this is one of the few examples, in my opinion, where the classical methods are still outperforming machine learning techniques in most cases. So that was one application which was widely used and is still being used. And then we realized at some point that you can actually even automate the naming of the new lineages. So if you remember, I told you examples of how the sublineage of XBB.1.5 and even the Omicron lineage, the B11529, was being named earlier by human experts, where they would first place the sequence onto the Usher tree and then see that there is like these long branches being incurred, and then based on that, they would suggest that a new name be designated. So they would make that proposal, and then some people would curate, and then finally an official name would be designated for that variant. This whole process could possibly also be automated, and that's basically what Jacob McCroom was also worked on. So he actually defined an information theoretic criteria where you can just view the tree, and from the tree itself, you can actually start identifying which nodes seem to correspond to new sublineages which might have emerged, and weigh those by the effect that those mutations could carry as well. So, for example, you want to weigh the mutations which lead to faster spread of the virus more than the mutations which basically end up doing nothing or could even cause harm to the virus. So that's kind of the idea. And what we found interestingly is that this particular software actually works pretty well, almost as good as what human experts would designate. So this is, for example, what these colors are shown by which parts of the subtrees would form different lineages. And as you can see, like uh, many of the branches which are colored differently are identified similarly between human expert system, which is shown on the left, versus our software AutoLens shown on the right. In fact, in some cases, you could even say that the automated system did more logical things where certain lineages that possibly should have been named and probably missed by human experts were also named accurately. Um, so this is a promising new direction, which we feel like uh, would take the field of molecular epidemiology even forward, and automate much of the processes which are currently manually handled, and also result in lots of time delays, right? Um, Similarly, the scale of this tree was so vast that we didn't even have good visual tools which could handle the scale of this data set. So way few, like almost no tool existed which could actually visualize all 16 million sequences at once. And a researcher at Crick Institute, Theo Sanderson, he actually developed this web interface, which I feel very inspiring work, where you can actually um, not just visualize the tree, but also interact with it in real time to study how the different strains have emerged and how the virus has been evolving pretty much in real time. Um, and then Alex Kramer at UC Santa Cruz again, he actually extended that particular web interface to not just view the tree, but also view the genotype. So what that means is that you can actually start seeing the sequence of each of these steps on the tree onto this particular interface. And so this is showing just a like the on the x-axis here, it's actually showing you the genomic coordinates of the virus, and here it's showing you different genes that the particular virus carries. This particular is a spike protein coding region, very important for this virus. And basically, what these colored bars indicate is the mutations that have been acquired. So, for example, this entire sublineage over here has acquired this particular green mutation at this position, right? So this whole sublineage has acquired it, and it's obviously common because they had shared ancestry. So many of these uh, closed sequences on the tree have these shared mutations as well. But of course, there are, as you can see over here, there are 30,000 such positions on the genome of this virus, and there are many tiny mutations that would be hard to even spot. So you have to zoom in order to see them. But there are, I would say, probably millions of or tens of millions of mutations um, uh, that are represented on this particular interface. Um, Next, Jacob also developed a very interesting tool called Cluster Tracker, which again I find as a very interesting application of this tool. So in many cases, public health officials, for example, would be interested in knowing like if a new strain of the virus or a cluster of related sequences circulating in a given region. So let's say in California, you are now seeing like a new strain that is like rapidly circulating. You sometimes want to know if that strain might have come from a traveler from outside of California and might have introduced that strain into California, right? So someone, for example, in Texas might have been infected first by that strain, and that strain might already be circulating in Texas, and then that person flew to California and then infected more individuals here, and now it's spreading like crazy, right? So those kind of applications are also very important, or insights are very important to health officials so that they know how to, uh, make some informed decisions, like, for example, travel restrictions and so on. Um, so what they would do in such cases is they would look at all the California sequences. So let's say those are the ones which are colored over here. And then they would see like around those sequences, for example, just at the root of those sequences, could they see sequences belonging to some outside region? So let's say these circled ones were from California and the outside ones were, these just outside ones were from Texas. Then they can actually infer that maybe there is an introduction that might have taken place into California from Texas. So this is what this cluster tracker is doing. It's currently doing this type of tracking only for the United States, the different states in the United States. So, for example, this is showing you that there is a cluster in California at this particular node of the tree. That's where it's circulating, about 900 samples from there. And it belongs to this particular lineage, XBB.1. And it also reports where it could have originated from. So here it's showing intermediate, but indeterminate, because it might be from many sources around it. But in this case, there's a New York region cluster which might have originated from California, and it's pretty confident about this particular inference, right? So that's what this particular tool is doing, and many of public health officials are actually using it in the United States, even as we speak, to identify and track these clusters. Another application which we, for example, developed in my lab with my students, as well as in collaboration with Rusk Orbit Datec, is that of ripples. And this is a tool which essentially identifies SARS-CoV-2 recombinants directly from the Usher tree. I started this work when I was a postdoc at UC Santa Cruz with Fresco Datec, and then my lab has developed some more optimization techniques after I moved here. And this work was published in Nature in 2022, mostly because it actually helped in carrying out the largest recombination study. So before I tell you what this like before that, I should tell you what recombination itself means in the context of the virus. So in some cases, what can happen is that one single individual might be infected by two different strains of the virus. So let's say one person caught an infection of the Delta variant from one of his friends, and then from another of his friends who was infected with Omicron, he also caught the Omicron variant, right? So what can happen with such co-infected individuals is that within that infected individual, two of those strains can hybridize. What that means is that it can take part of the genome sequence from the Delta variant, part of the genome sequence from the Omicron variant, to form a completely new strain, which is like a hybrid of both, right? So you can call it Deltacron if you like. And those type of variants do exist in the nature, but Why they are more concerning is because with this, it can actually add a lot of genetic diversity in a very short span of time and can acquire mutations which might be much more advantageous. So in a sense that it can be more contagious than both of the individual variants like Delta and Omicron, with a giant leap in its fitness, right? So people do care about like these hybrid variants because there's always a concern that they can be far more infectious than just normal speed at which the virus is acquiring mutations, right? So that's kind of the idea. So in general, detecting these recombinants is a very, very hard challenge from genomic sequences, especially when we are talking about scales of millions of genome sequences, right? In fact, most tools that were developed for recombination inference were only able to handle a few hundred to few thousand sequences at once. Uh, so millions was pretty far off for them, pretty out of reach. But for that, what we did was we actually uh, developed a software which takes advantage of the fact that when you have new variants emerging, they actually result in really long branches in the tree. And those long branches tend to be rare. And the reason why you have long branches is because they neither look like Delta nor like Omicron. So they are dissimilar from all existing sequences. So they result in like lots of mutation on the branch at which they were placed. So you can just focus on those long branches and ask this question if they could be better explained by sort of a recombination event of two other existing sequences. So in this case, we show an example of that, where this particular sequence that appears at the root of this tree looks like a hybrid of two other sequences that existed earlier, right? So that's kind of the idea. And so this is how Ripples was developed. And like I said, it did a comprehensive recombination study of SARS-CoV-2. In fact, it's able to complete this analysis now with all the optimizations that we've done in a matter of 4 to 5 hours only for all sequences combined. And what was one of another important observations that we made was that the breakpoints, where the points at which they are hybridizing, they seem to be more prevalent in the spike protein coding region of the SARS-CoV-2 virus. So that's what you're seeing over here, that they occur at a higher frequency here than other regions of the genome. And there's no good explanation for this, but it's an observation that intrigues many. And some others have also observed something similar in the context of past coronaviruses, that this does happen. Um, and then another undergraduate from my lab, Carl Smith, he actually developed a way to visualize these recombinants as well, which I really like this particular interface. So this interface gets updated every month or so currently, where we do a reanalysis of recombination of using all the sequences that exist till that point, and then we display a big table which shows like where in the tree do we see possibilities of recombination and which strains of the virus might have recombined. And this is a good example of how we visualize this. So in between, we are showing the recombinant sequence, not all positions of it, only some of the important ones highlighted. And then these are the two parents which might have recombined, the acceptor and the donor. And what you can see, I would suggest you just focus on the highlighted positions here with blue and orange. And what you would see is that it's very likely that a breakpoint might have occurred here because in this region, because prior to this breakpoint, it seems like the recombinant sequence looks mostly like this blue sequence, which is that of the acceptor, this one. And after the breakpoint, it has mutations which mostly belong to the donor sequence, which is shown on this orange highlighted mutations, right? So it changes its appearance at certain point in the genome. So it's quite likely that a big point might have occurred here, and this is where the hybridization event might have taken place. So this is sort of the visual analysis that people can perform, or those who are interested in studying the recombinants in more detail through this interface. And in fact, it can be used to identify recombinant strains as well. So that's pretty much it, what I had planned in mind. I would just say that we are actually extending Usher to many pathogens. In fact, we've done that already for monkeypox. We've done that for RSV, influenza, avian flu, and many others. And we are also going to soon release the tuberculosis strain, which is a bacteria. And also we are doing further optimizations to make sure that we can handle ever-growing scales, but also more diverse pathogens. And we remain to be super excited about this field as we feel that it's very impactful. but also a great mix of computer science applied to real biological problems, which has real implications on human health and public policy as well. So with that, I'll end my talk, and I'm happy to take questions.
Awesome. Yeah, thank you so much. That was a great talk. Hopefully that helps clarify some of the intersections between biology and engineering. I don't know, any questions from folks? Feel free to type in the chat. Feel free to unmute. I have one saved up if not.
I have a question. Um, can you discuss your experience with handling and manipulating large genomic data sets, including strategies for efficiency and optimization?
Yeah, that's a great question, and I love the fact that it's so broad as well. So I love this, field because with genome sequencing data, you are essentially seeing this exponential trend, which has just not stopped for at least 2 decades now, where uh, it's by exponential, I basically mean that it's doubling every fixed interval of time. So let's say a year or so, it could double, or 2 3 years, it could just double in volume. What this means is that this field is creating like a very dire need to keep innovating, right? Because what you developed, let's say 5 or 10 years back, might just not prove to be sufficient to handle the scale that we are seeing now, right? And this problem becomes even more severe given the fact that our processing capabilities, right? So I actually come from a more computer engineering background, computer architecture background, where the field was all about how do we increase the speed of computers, right? So how do we change the architecture of computers so that they can process it faster and the transistors keep getting smaller and more efficient? And in recent years, that has become a big challenge, right? So it's becoming even more challenging to increase the speed of computers. So what I mean by that is the tools that you develop today are not necessarily going to get faster just because of the fact that your hardware got faster, right? So the software now has to innovate as well, uh, in tandem with some of the hardware speedups that we are still getting, very small amount, but we are still getting it. So the tricks that we use is that earlier, and the bioinformatics of, let's say, a decade or so back was all about this, where the focus was more on how do we do one task very efficiently, so a single task, right? So the way programs would be written is that you just have a stream of instructions and you do it one after the other, right? You process them one after the other. And the new paradigm, if you really want to extract speed, and that's basically has been our secret sauce in many ways, is to move it to parallel computing framework, right? So there, the idea is that you don't have one stream of instructions done sequentially. Rather, what you do is you have parallel stream of instructions. So what that means is many of the things are happening at the same time. Many data is being much of the data is being processed in parallel, in tandem. And so, because more work is being done simultaneously, you actually get much higher speed ups. And in fact, this is the way in which fields like AI, for example, are also progressing, because uh, it's actually newer processors like GPUs that you might have heard of, like companies like NVIDIA and AMD build those. And these devices are inherently parallel. The reason why they are so fast is because they are parallel. So many things are happening at the same time. But the trick here is, or the difficulty or the challenge here is, how do we parallelize your computation, right? Because we generally are engineered to think very sequentially, right? Do this, then do this. We like to give instructions to the computer in a very sequential fashion. So what we do in our lab is to train parallel students to do parallel thinking as well, where the idea is that you identify where in your program can you actually do things in parallel without affecting the correctness of your program, right? So have multiple instructions executing in parallel, which can take advantage of the parallel computer architectures like GPUs or multi-core CPUs, or in some cases even parallelize them over many machines at the same time, right? Many independent machines that can communicate with each other. Uh, how do you parallelize the computation? So that's kind of the idea, and that was true for Usher as well, where we actually handle a lot of parallelism, in the sense that when we search the tree, we actually search every node of the tree in parallel, right? Where could it be placed, and evaluate whether this is a good good location to place that new sequence or not. That whole thing is done in parallel. So that's one example. Then there's another more fundamental way as well, and Nima is much of an expert on this topic. He teaches advanced data structures, but those data structures tend to be very important in our work as well. So they determine the efficiency. So by data structure, what I mean is, how do you represent the data in a computer, right? How do you represent the data that you have in a computer so that you can compute on it very efficiently? So in genome sequence processing as well, this tends to be a very important problem, although you might think that these are all just genome sequences, so there must be one way to represent it. But depending on what you're doing, there might be more efficient ways of representing the data that you're processing on. So just to give you an example, I told you in Usher, we developed this data structure called MAT, mutation annotated tree. And the cool thing about this data structure is that it's much more compact than storing all sequences that it represents. That's the cool thing about it. The reason why that is the case is because we store one sequence at the root and just a bunch of mutations that led to other sequences. And that tends to be much more efficient to store than, have every sequence represented once. And most of the information in those sequences is redundant because they are actually very similar sequences. The other reason why it's so much more efficient to have this data structure is because once you have a MAT, finding the location where to place a new sequence on the tree becomes very efficient because all you have to do is you already know the existing mutations up to a certain node in the tree, and then you can just identify how many new mutations do I have to add to accommodate my current sequence, right? The sequence which I'm trying to place. So it becomes more efficient to do that, and the output that you get is also a mutation annotated tree because you've done the computation necessary to accommodate the new sequence, and that representation is also MAT. So you can keep doing this essentially. So the idea is that you don't have to process over and over again the raw data, which is actually the genome sequence data. You've already pre-processed it into an efficient representation that can be used for this particular application, like phylogenetic placement. So we do a parallel research in both, simultaneously research in both. Parallel computing, like how do you design algorithms which are inherently parallel, and then how do you represent them, the data that you have, very efficiently. And that tends to be much of the secret sauce that we have.
Great question and great answer. Awesome. Any other questions? Feel free.
Can you guys hear me? Because it's
Yeah.
been interesting. Okay. My last question is, how do you ensure the reproducibility and accuracy of your computational system analysis?
So if I get your question correctly, by reproducibility basically means that if you were to do the same analysis that I did, you would be able to get same answers, but potentially that's kind of the essence of the question, if I'm not wrong. Clarify if needed, but I'll just assume for now that that's what you meant. So in our case, like the algorithms that we design are actually deterministic in the sense that if you give it the same inputs, it will actually get you the same output. So it's done by design, although it's not a necessary in some of the algorithms that you need to have deterministic outputs. But in our case, at least in this example, it is deterministic. So what that means is you do it over and over again with the same inputs, you are going to get the same outputs. We also have essentially released all the codes, and like the software is publicly available where people can actually install it, try it on the data sets that they have, and try to reproduce whatever we've done. All the instructions are there. In fact, even the script for the maintenance of the Usher tree, right? Basically the script that we use to download the sequence from public databases and then place them onto the existing tree, all of that is also publicly available. So people can actually use the same script, do exactly what we are doing to maintain the tree if they want to. That's actually open sourced again by Angie. Her repository has all of this information. But in addition to that, we also release the data that we are maintaining, like mutation annotated trees are publicly available. So people can download those and do further analysis on them if they like. But it's a very important question. Reproducibility is a very important issue generally in academic research, and it's a standard that should be maintained. So I'm pretty glad that you raised that question, and I hope I answered it satisfactorily.
Yes, Thank you. I do have one more question. I'm sorry. I know you said that you have come from like a background with engineering, but what specifically, any other reason for you to want to pursue like electrical and um computer science? I don't know if that makes any sense, but what made you specifically want to do that
Like a
besides
mix
your
of
background
electrical
with
engineering and
engineering?
computer science. Is that
Yeah.
what? Okay. So actually, within electrical engineering too, I was more involved on the computer engineering side. So what I mean is that I was always interested in um, fields like computer architecture, like how do you build like new processors, like I mentioned earlier, right? Design the architectures of processors which make them faster, more efficient, and so on. So it was more on the hardware oriented side. So electrical engineering, much of the electrical engineering is actually very hardware focused. But of course, like computer architecture is as close to software as you could be, right? Because it's right at the interface of where the software begins, right? Because it's at the architecture level that the software gets to interact with, right? And computer science in many ways is uh, more closer to software, and that's where it's higher up in the stack than electrical engineering as a field is. But they are actually very related fields. There's a lot of overlap between both these fields. So it's not very hard to switch between the two. If you are from one background to, for example, in computer science, if you want to move to electrical or vice versa, it's not very difficult to do. But the sort of interests that I have, they naturally fall at the intersection of the two fields, right? Because as you saw, we are very interested in hardware design techniques as well. How do you build new processes? But actually, much of my work today, including the ones which I presented, is a lot about software engineering skills, about data structures, and so on. Sort of the stuff that Nima also teaches. Uh, so I feel like these two are very like they are very rightly called sister fields. So computer science and electrical engineering. In many schools actually they are one and the same department. Like in Berkeley, for example, they have ECS as one department. MIT also has ECS. So I would say that they are not as different as some other fields of engineering could be.
Okay, thank you.
Awesome. Thanks so much. Yeah, thank you again, Yatish, for joining us. That was a great talk, and thanks for answering our questions. And yeah, I really appreciate it.
Thank you so much, Nima. Was great to be here.
Awesome.
Enjoyed the interaction.
Yeah, have a great rest of your day, everybody.
You too. Bye bye.
Bye, everyone.